Okay, so welcome to this VEuPathDB webinar. I'm Suzanne Warrenfeldt, part of the outreach team here, and I'm joined by my teammates, Sam Rund and Gloria Geraldo Calderon from the University of Notre Dame, and Eve Basinko from the University of Liverpool. We're here to fill you in on the new features and data in Release 53, and to demonstrate how to access and interrogate some of that data. You can ask questions and as we demo things. In your control panel, you should have a questions panel. If you have a question during the webinar, you can type it in there and we will try to answer that as soon as possible. And sometimes we will stop for discussion. So this webinar is being recorded and you will receive an email tomorrow with a link for the recording. But we also post those to our sites. All upcoming and recorded webinars appear on our webinars page. I'm showing the VEuPathDB site right now, which is our portal to access data across all organisms we support. To get to the webinar page, you use from the header, go to the help menu, learn how to use VEuPathDB, and this webinars button in the middle will get you to the webinars page. And I will copy that and put it into the chat here. So if you don't get your email or you can't find it, you can go to this page here. It'll be updated tomorrow with a recording of the webinar, which will appear in the previous webinars. And all of our component sites have the same look and everything is located in the same place. So if I want to find the webinars page on CryptoDB, for instance, I would do the same thing, help and learn how to use VEuPathDB. So I'd like to begin by mentioning the Release 53 features that appear on all sites. And this is detailed in our news. Every release that we put out has a release news associated with it. You can reach that over here in the left side panel of the homepage of any of our sites. And I'm just going to go to the Vectorbase Release 53 news. And look at this new features in Vectorbase 53. So they're all detailed here. I'll just go through them really quick. We have a new Reddit site that we share with our other bioinformatics resource centers. You can reach it from the header of any of our sites. There's a link here. You will find posts and conversations that concern not only our sites and organism, but those of our colleagues in the other bioinformatics resource center that work on bacteria, virus, and flu. So you can join the conversation here with us if you like. And then feature number one is that our dataset records have an improved format to increase clarity. I'd like to show you the genome record for Plasmodium falciparum 3D7. It's a very well represented organism with a lot of curation and a lot of links. So if we can, what I've done is gone to PlasmoDB, opened up the data and looked at this page, data sets in PlasmoDB. Now it's showing 276, but I'm going to filter that based on 3D7. And I can find under the genomes category the dataset page for the genome sequence and annotation for this organism. At the top, then we have a summary of information followed by further below, we find a navigation pane to sections that describe the data set, and this the data set release history table is very telling and has a lot of information. It chronicles the changes that we've made in each release of this data set. We have link outs. This is very interesting too, and very useful. So this is categories of other data that are aligned to this genome. So if I look at RNA sequence data, there are 23 data sets that are aligned to this organism, and this is a link to the data set page it says 24 to a result that returns the 24 RNA sequence data sets that are aligned to PF3D7. You know, and other data too. You can quickly get to the publications, and from there I can actually go to other data sets, added data set pages to look at the details for that. So the let's stay on PlasmaDB. The site search now accommodates, I'm sorry. The site search up here in every header now accommodates versions of an ID. So you can search using the ID without an extension to retrieve any occurrence of the ID with extensions. So let's say that I am looking for a protein. I'm looking for the gene that is associated with this protein. I got this protein ID from NIH, NCBI. And if I search without the extension, I am returned 2 genes. If I go to that record page, I can see that in the aliases table, that ID is actually present as a versioned ID. So we've broadened the search. You don't have to search exactly for the IDs to be able to find results in our pages anymore. And the other features that we've added concern Apollo. Let me go there. I did tools and Apollo. I'm presented with a landing page that has help information, and now I'm going to go to Apollo. So Apollo is a community annotation platform. We welcome community input on gene models and offer Apollo as a way to record your user suggestions. We've updated the Apollo version to 2.6.5, and we've added a BLAT feature to help in finding the genes you want to annotate. So you can use this search panel here. As soon as it loads, we can use this search panel here where you can enter a nucleotide or a protein sequence. When you do that, you'll retrieve some links that will help you help locate the sequence that you want to annotate there. We're not going to go into any more detail on that. But those actually are all of the features that we've added into this release. So now I would like to turn it over to Gloria, who will tell us about new data.
Thank you, Marianne. Oh, sorry, Suzanne.
Yeah.
I'm going to change presenter. Okay, show my screen. So you're looking at my screen now. Is that right?
Yes, we are. Thank you. Yes.
Thank you, Suzanne. Hello, everybody. I'm very excited to tell you that for this release, and as Suzanne already showed you, go to the side panel and click here on news. We have 13 new omics datasets. So for the first time in our new website, we have in the same release genomes, transcriptomes that come both from RNA-seq and microarrays, and proteomes. We have mass spectrometry data. For the genomes that we have for this release, we have 2 Anopheles mosquitoes, and we have 1 tsetse fly. For the Anopheles species, the strain is going to be changed in the next release, and this is going to be the new strain. This was a request from the data provider. This other genome that we have right here at the moment has no annotation because there was an issue with an INSDC record, but for the next release, it will have it. For the RNA-seq data, we have bite niches, and we also have data from mosquitoes. From the microarrays, we also have data from mosquitoes. And today, what I'm going to focus on is on the proteomics data sets. And I just want to tell you that with all these new data, which as Suzanne already showed you, you come to data sets in VectorBase, we are reaching almost 500 data sets available for you in VectorBase. And as always, if you want to suggest us new data sets to load here, click on contact us and let us know. You don't have to submit twice, just give us the accession number from the INSDC record, and we will take it from there, except if we are talking about proteomes, in which we do not take raw data. So just send us a spreadsheet with the gene IDs and the peptides, and we will take it from there. But as I said, contact us for more detail. Okay, so these are all the data sets that we have in VectorBase. And if you only want to see, for example, the proteomic data set, so you can say, oh, well, for the category column, which is this one here, I just want to look at what do you have in proteins. And because we just loaded 2, so that's what you have right there. So you can click in any of these 2 links to get more details about that data set. So this is similar to what, sorry, that's not the one. This is similar to what Suzanne was showing you. She showed you the page for the genome. I'm showing you the page for the proteome, and it will have the link to the publication, a summary that we wrote for it, a little bit, a 1-paragraph description giving you more details, the link to the publication, and what you can do with that data set within VectorBase. And if we have external links, we will show that too. This is something something that Suzanne, I don't know why today I want to call Suzanne Marianne. Sorry. So something that Suzanne showed you also is the genome pages that you can, in addition to be able to get to them from the data sets in VectorBase, you can also get to them to the organism link. These are only the genomes. And now the way they are structured, they are more similar to what legacy users used to remember as the organism pages. So that will be similar to you guys. Okay, so now I'm going to demonstrate the proteomic data. So if you come to, as Suzanne said, the layout from all the pages is the same. So if you click on proteomics, you have 3 different types of searches, and this is true for other sites. In VectorBase, because we just started, if you do the same thing, you're only going to get 1, the mass spec evidence, but we do have the capability to load any type of data set, so just let us know. So if I click in this search, mass spec evidence, I will arrive to this page, identify genes based on mass spec evidence. The first thing that I get here is that I'm able to select the species and the experiments. So I'm going to expand them all. As I said, we only loaded 2, one study from 2013, this is a study from 2011. And you are able to select all the data from that species, or you could select, for example, only one of the samples there and like this. So for this demo, I want to select everything. But once we have more data, you could say, well, and it's difficult for you to find it reading the table, you can, the list, sorry, you can just start typing words and say, oh, I only want midgut data, and that's how you can find it. Okay, I select all of this. Here, The minimum number of unique peptide sequences is of course going to be 1, but the higher you go, the more stringent your search is going to be. So for example, if I say 2, so I want 2 unique peptides for my genes. And here you have per sample or across samples. So for example, this means that if I have selected all these samples, so those 2 peptides can come from any of these samples if I have selected across them like this. But if I have said per sample, each one of these bullets needs to have the 2 peptides. So I'm going to say per sample, but I don't want 2. I want to be very stringent, and I want 100 peptides. There is some advanced parameters here that you can also play with. And for this, as for any of our other searches, we have a description that tells you each one of these parameters, how to set it, what do you expect, this is, which is what I just explained to you. And these are the links to these proteome pages and their publications in PubMed, which I just demoed for you. Okay, so I said this for all the samples, so 100 peptides per sample, get answer. So I had run this earlier, so that's why we have it here. And this is also to tell you that I have recorded this in the page. And here, when you have the public strategies, you can come and load this strategy to know what I have done. Okay, so you come here, you can give it a name, you can save it, same as with any other. And I obtained 12 genes. You know, these are all coming from Anopheles gambiae, so I don't need this panel here, so I can hide it if I want. The first columns are the same as for any other search in our websites. You have the gene ID, the transcript ID, the location, and the product description. These 3 columns at the end are the ones that are unique for the proteomics data sets, selected samples that match, some of the unique peptides and unique peptides. And you can sort the columns like this, or you can say only, you know, show only 1 transcript per gene, or show them all, and how many rows you want to show here. And you can click on one of these genes to look how the proteomic data looks in the context of that gene only. And here, I was going to show you the links of what the data was about. So if, no, sorry, what did I, okay, no, never mind. I was just going to tell you that when you click on the publications, you get to the PubMed links. Okay, where I was here. Okay, so if you click on one of the genes, you go to the gene set page and you load that. And for example, you will arrive here. This is the gene page. And let me go to the top. This is what you will see at the beginning. And with this navigation panel here, just as Suzanne showed you, you can go to any section. In this case, I am interested in proteomes. And I go to the mass spec evidence section. And here, the first one is just a table showing me for each experiment and the samples, how many sequences do I have. The second section of this proteomics area is you have the possibility to go to the genome browser, to the protein browser, because we have 2 browsers. So here at the top, you see the genome browser. But here, what we are looking at is the protein browser. And you can interact with this from here. So you click on scroll and zoom, and you are able to interact with it. You can close the tracks, or you can click here, view in protein browser. And when you do this, you can select more tracks. Here, for example, I have selected for you all of the ones that we have available. Click on back to browser, and you have transmembrane domains, interpro domains, in addition to what you already were looking at when we were in the gene page. But you could also click on the Genome Browser. And what we have here is just this single protein. If you go to the Genome Browser, you go select tracks and select all the available tracks that we have for proteomics, load them all, back to browser, and you have, this is a combined evidence, not for a single experiment like we were seeing before, but for all the mass specs that in this case is just 2 experiments. And that's all I wanted to show you guys. I don't know, Suzanne, did I miss something?
No, I think that's good. So just to give a recap, Gloria searched a certain proteomics data set. She came up with a list of genes, and she went to that gene page to interrogate the proteomics data there, and then transitioned to visualizing that in a browser by using the buttons in certain sections on the gene page. And that shows the trail workflow of how different tools are integrated in our sites. That's great.
Thank you, Suzanne.
Yep. So I think we're ready to move on and change. Sam Rund now will talk a little bit about new data in our MapView tool on VectorBase.
Thank you, Suzanne.
Yep.
Yes, I'll be showing you some updated data that we've added to MapView, which is currently only accessible in VectorBase, but hopefully soon we'll have data from our other sites integrated into the map. So again, I'm going to do what Gloria and Suzanne did and start from the release notes for VectorBase. And if we go down here to population biology, which is our MapView projects, we have 7 totally new projects that were added in 22, where we just did some updates or maintenance to clear up any errors or that may have appeared. And I will be showing you 3 different of these 7 studies, because they all involve a different kind of data, but it's all going to be geospatial. So the first one I'm going to do here is a paper from Cameroon, and this is reporting on insecticide resistance in Anopheles Glutathione mosquitoes. And so this would be an insecticide resistance phenotype. So I'm going to view on the map. Open the new tab. Let's see if I can do this. And so here you can see the data, and this data type, this project actually does have some genotypes, but since it only has one, I'm not going to show you that. So I'm going to click here, and you'll notice that that switched to this insecticide resistance view. We have different views. Most projects or papers will only have be accessible from one view. So now we're in the insecticide resistance phenotype view, as I mentioned. And I can click on this data point and see a graph of the data. Now this looks a little odd because our system generates background by taking all the data for the point that you click on, and the background is all of the data that is not shown, that is not selected. But in this case, we're only showing one project, so the background and the selection are the same. But I will take that off in a second and show you what it looks like. And so you can see for each of the data points we have, you can see the date, where it was collected, the test that was done. So here was bendacarb, 0.1% concentration for an hour. And you can see that this sample had very high mortality, 90%, these at 100. But for example, they were very resistant to DDT. So DDT at 4% for an hour, these mosquitoes seem to be almost completely resistant to DDT because the mortality rate was only 4.16%. And if I just briefly get rid of this X, you'll see all of the other phenotypic data that we have. So even from that location, you can see that now our number went up from 8 assays to 148 assays. So you can see a lot more data. And we can also do things such as start to add filters to filter by, say, just Anopheles gambiae, and perhaps we want to say it loads, I'll pick a specific insecticide, so 2-deltamethrin. And so you can drill in and find the data that you want. And we do see that in this area, there are some very susceptible Anopheles gamete that have been recorded, and some, again, some very resistant ones. So, okay, so I will go back here, and now I will select a, so this data set that's new is another year's worth of data from the, I'm gonna go this one. Another year's worth of data from Marion County, so that's Indianapolis. It's my hometown. And so we'll view this data, and this is abundance data. So instead of analyzing insecticide phenotype, we will look at a population surveillance. And again, I'll click here, and that will switch us to abundance view. And you'll also notice that here we have a lot more locations that are collected. And I can click on a data point. I'm going to rechange the colors so it's easier to see. And you can see here the seasonality of this data of the collections of mosquitoes from that location over the season. And we can also zoom out, and it will re-aggregate some of those data points, and the graphs will adjust to the new aggregated data. Now, I will note that, for example, for places like Marion County Health Department, so even though this release is just an extra year of data, if I instead of putting that project ID, I put the depositor, we can see that data for Marion County and many other places on our map for population surveillance actually goes back well over a decade. And the last one I will show is this study from Anopheles arabiensis and Melis in Senegal. And this is a study of insecticide resistance genotypes instead of phenotypes. So if I view that on the map, and I'll do insecticide resistance genotype, so that changes the view here to genotype. And we can see here the breakdown, the allelic breakdown of mosquitoes collected with the 3 different alleles of the KDR knockdown mutation. And again, I can always click this button here, list details, to see all the different metadata that's available for each of those collections. So for example, in one collection, 91 individuals were collected and 65% had the had the wild type mutation. Whereas here we have a 9% and then were the L to F knockdown mutation. And that is all.
That's great. Thank you. It was really neat to see the difference between all of those different data sets and the type of data collected in each of those data sets. It's really seems like a very powerful tool. So we'll move on now to Eve, who is our fungi outreach person, and she'll demonstrate some of the new data in 53 there.
All right, thank you very much, Suzanne. So you are now viewing FungiDB news release page, and in this release we have integrated various data sets, including genomes of fungal pathogens, for example, Blastomyces, Coccidioides, Fusarium, Malassezia, and also important model systems such as Cluromyces lactis and others. If you click on one of the links in blue, you will be redirected to a dataset record page. And as Suzanne and Gloria have shown you before, this is a page where you can learn more about relevant publications, genome version, and also learn about all available searches that can be deployed for this particular dataset. For example, if genome is annotated, you will see that identified genes based on different criteria will become available. However, if genome is not annotated, it will be limited to genomic sequences and genomic segment searches. So coming back to the news page and scrolling down, you will also see that in this release we integrate lots of functional data, and that includes Snip datasets, RNA-seq, chief-seq, proteomics, and we have also updated functional annotation for selected organisms and also Go terms. Go terms are used by Go enrichment analysis, which is a powerful method to analyze gene lists that are returned by your searches. And if you would like to learn more about how to set up and run Go enrichment analysis or different type of analysis in general, check out our previous webinars, and they can be located under Help, and then Learn how to use VEuPathDB. Okay, so now we'll switch gears a little bit, and I will go back and demo how you can create a two-step search strategy using newly integrated data sets. And in this experiment, in silico experiment, we will look at the Snip dataset that was collected, where whole genome sequences were used for calling SNPs. And this dataset deals with clinical isolates where patients that were infected with Cryptococcus neoformans were undergoing antifungal treatment. And the second dataset that we'll take a look at, we'll actually look at which genes are being downregulated where you delete an important transcription factor. So just coming back a little bit to summarize as to the type of the data that we are going to look at, I'll bring up a PowerPoint, and give me just a second. So, and the PowerPoint is right here. So the in silico experiment that we are going to set up, as I have mentioned, deals with two different data types. And in the first step, we will find genes that have non-synonymous SNPs. Non-synonymous SNPs are known to have a role in the emergence of emergence of the resistance to fungal treatment. And so in the first step, we will ask a question by saying, how can I find genes that have at least one non-synonymous SNP using the clinical isolate study by Stone et al. 2019. In the second step, we will actually explore the RNA-seq dataset that have looked at multiple genotypes in Cryptococcus neoformans, published by Homer et al. In 2016. And we will find genes that are downregulated in GAT201 deletion mutant. And why this is important, GAT201, 204, and LIF3 control a transcription factor network that has overlapping targets, but both 204 and LIF3 are the targets of the GAT201. And all of these factors have been previously implicated in virulence. So the question that we are essentially will be asking, how many of the genes that have non-synonymous mutations, at least one non-synonymous mutation, are also downregulated when you delete GAT201. Okay, so let's come back to the new section and navigate to the data set that contains SNP records. And so I already opened this here. And as I have shown you before, if you scroll down, you'll be able to see the available searches that can be deployed for this data set. And now we'll go ahead and click on the identified genes based on SNP characteristics. Now, I have previously pre-run this search, so my parameters are being remembered. But if you are first time setting up the search, you will need to select H99 from pull-down menu because we're working in Cryptococcus neoformans H99, and select a dataset, which is this one that was integrated in release 52. And now there's only two things that we need to do, is to select the class of SNPs that we are interested in, such as non-synonymous, and set the parameter for the number of SNPs in the above class to at least one. So this ensures that the SNPs that you will return in the next step will be non-synonymous. If you change this to 0, you will be essentially returning all of the SNPs as shown here. But we want only non-synonymous, so I'll go ahead and select parameter for 1 and click on Get Answer. And so when the page refreshes, we will be able to see the search strategy that we are starting to create. And I will go ahead and rename the first search just to kind of keep in mind of what type of records we are interrogating. So non-synonymous SNPs. Okay. So here we are. And I'll scroll down to take a look at the list of genes. So in summary, I have slightly over 4,000 genes that have at least one non-synonymous SNP based on the clinical isolates collected by Stone and colleagues. Now, the next question that I want to ask is, well, how many of these genes are actually downregulated when you look at the RNA-seq evidence from the GAT201 deletion strain? And so what I want to do is essentially combine two different types of data sets. And you can do this by clicking on the Add step, and then combine with other genes will remain the same because we are looking for the genes. And we want to intersect our search with another data set. And in this case, I will use filter to start typing RNA to bring up RNA-seq evidence page. And this page lists all available data sets in FungiDB. However, I will quickly filter on H99 to bring up anything for Cryptococcus H99 and scroll down until I see the data set that I'm interested in. And here is this data set right here. I'll choose fold change, which will look at the fold change within the specified criteria, such as I'm looking for downregulated genes, and notice that this data set is trans-specific, but by default I will leave my search at the sense strand search. And so I'm interested, first of all, looking for downregulated genes. So let's go ahead and set up our search. There are lots of different strains and it looks a little bit busy, but the main difference is that there are two strains involved. KN99 is a lab strain and CM18 was collected from a patient. So we are interested in using CM18 strain and also the GAT201 deletion in its background. So for the reference strain, I will use CM18 unmodified strain, or essentially for this purposes, it will be wild type control. And for the comparison sample, I'll be looking at GAT201 deletion at the same OD as the control sample, okay? And notice how the graphics on the right changes to represent graphically the type of the search that you select and the type of the results that will be returned once you click Run Step. So let's go ahead and do that. And I will go ahead again, rename my search step to remember the genotype that I am exploring in this case. All right, now, so here is the result from our two-step strategy. And so notice that there is a list of genes on the bottom that you can scroll down and explore. And if you want to look at the graphical representation of the transcriptomics data that we had just looked at, you can also click on bring columns, or add columns rather. Look for the, for example, Homer is the name associated with this data set. And in this case, I will go ahead and bring up the graphs that showing both strands TPM graph. And so the view will refresh, and now you have a list of genes and also a panel of how these genes respond within that or the other treatment. So the summary from this analysis is that I have identified over 4,000 genes that have non-synonymous mutations. And for me, it's interesting because I know that non-synonymous mutations may be involved in the emergence of antifungal resistance phenotypes. And I have also looked at the particular deletion strain that is known to control a transcription factor network involved in virulence. And so in this particular background, when GAT, or the master sort of commander of GAT 204 and LIV3, is deleted, I have 393 genes that are downregulated, but only 277 of those genes have both at least one non-synonymous SNP and downregulated. And so there's a lot of things that you can do further on to continue analyze your data in silico. For example, you can click on Add Steps and combine with other types of data. You can also transform this data into orthologs in other species, and that could be a use case where you work with Cryptococcus, let's say Neoformans JC21, but these type of records are mapped to the H99 reference genome. So you can essentially convert the your findings in this experiment into JC21 by running an ortholog transformation approach step. And you can also combine other types of data that are non-gene lists by using genomic collocation tool. As I have mentioned before, GoTerm enrichment analysis is also quite powerful in analyzing your results. For example, if you click on the Analyze Results tab, and you will be redirected to a view where you can pick how to enrich your analysis further. And for example, we can click on gene ontology enrichment and then look at the GoTerms that are enriched within the 277 genes returned in my step 2. And so for the purpose of this demonstration, I'll use biological process and then click submit. And so the page will refresh in just a few seconds. And so while we wait, I'll just go ahead and mention. So this analysis is quite powerful when you try to look at the genes that are enriched in your results. However, there is also a caveat that if you have hypothetical genes, and we can actually take a look to see whether we have any within our results by scrolling down. So if I don't see it at the moment, but you could imagine that your result may return hypothetical genes. So in this case, if these type of genes are not annotated with GoTerms, they obviously will not be enriched or brought into your analysis. So you may want to devise a search where you will look at the hypothetical genes independently of Go enrichment analysis to learn more. But looking at the GoTerm analysis here, I'll go ahead and sort of on the genes in my results that have the highest numbers in terms of the GoTerms annotations. And it looks like the genes that I have are enriched in terms that are relevant to response to various stimuli, stress, chemicals, oxygen compounds, etc. And to me, it seems probably appropriate for the type of the data that we are looking at, because first we have looked at the SNPs in clinical isolates where patients were treated with antifungal drugs, and then second we looked at the genes that are specifically downregulated by a in the background of GAT201 deletion strain, where GAT201 is a transcription factor that is required for the response in virulence. So here it is in a nutshell, a very brief demo of how you can create searches within FunGTB and how you can also bridge different type of data sets to address biologically relevant questions. And Suzanne, I believe I will pass it back on to you.
Okay, great. Yeah, thanks for that demo. It's really wonderful. So Eve went into more depth about how to combine searches and not just do a single search, and to look at the biological characteristics of or determine, a list of genes that have certain biological characteristics that you're interested in, which is really great.
And by the way, if I can just mention for a second, this strategy is available in FineGTB under public strategies. So you can just navigate, click on the link, and you'll be able to display it on your screen.
Yeah, that's great. Okay, so I will be outlining the new data in the Parasite databases. There's quite a bit of new Parasite data in release 53, and it's spread across several sites. So I created a few slides to summarize that data. And I'll take some side steps to from the overview to demonstrate some of the data on our sites, and I'll we sort of have a concentration on looking at Snip data here, because I'll demo some of the Snip and RNA sequence data in the parasites. So in AmoebaDB received one RNA sequencing data set. It is the transcriptome of Entamoeba histolytica in normal and stressed conditions. And their stressed condition was serum starvation, and their normal was control culture and a serum replenishment. CryptoDB actually received 3 data sets, all whole genome sequencing of isolates, the type of data that we analyze to determine single nucleotide polymorphisms, or SNPs. So the first one had 4 isolates that are C-parvum laboratory strains. The second one, second data set, had 15 C-parvum isolates from animals, and the third one had 7 isolates from humans. And here's where I'm going to diverge from my overview and go in a little bit more detail into the ways that you can interrogate this data. And the first way is something that is a search that Eve just showed, which I'll repeat, and that is to investigate genes with SNPs represented in these data sets. And then the second way is to investigate SNPs that are common in these data sets, but not necessarily associated with genes. So what I'm going to do is look for genes with SNPs common to the animal isolates. So I will go to CryptoDB and look for that search. I'm going to look for genes based on SNP characteristics, and I'm led to this search page. I know the data set that I want to look for is a C-parvum data set, so I'll choose that parameter. And now my list of isolates is present in this set of samples parameter. And I can actually use this parameter to group any of the isolates, any of the 73 isolates together, and call that a group that I want to determine SNPs in. So for example, if I knew that I wanted to look for, to analyze for SNPs only that were found in human isolates where the host was human, I could choose this data set here, and I would be determining SNPs in only those 43 isolates. But my objective here is to look at isolates from animals, this new data set that we've integrated. And so I am using the metadata parameter data sets. And on the right, I have details of all of the data sets. I'm going to choose the new one that we just integrated, C-parvum isolates from animals. And then I'll choose to look at non-synonymous SNPs, and instead of looking at all, I'll choose between genes that have between 1 and 5 non-synonymous SNPs from this determined in this set of isolates from animals. So I'll click Get Answer. And I see that I'm returned 860 genes that have between 1 and 5 non-synonymous SNPs. And so let's say I'm interested in this first gene here. I'm going to click on that, go to that gene record page. And I'd like to demo this excellent tool that we have. It's an isolate alignment tool. So on this gene record page, I can use the contents section or this handy dandy button up here to go to the Snip section. I get an overview of all SNPs for this genes in all isolates, not just the one that we searched. And then there's this great tool here, isolate alignments in this gene region. And so I can actually retrieve a multi-FASTA sequence of any of the isolates here, or I can produce an alignment. So let's go ahead and try and produce an alignment in this gene region for the isolates that we searched. That's going to open a new page and run our alignment. Here we have our results. It used Clustal Omega to produce an alignment. Here's the alignment. You can also view a phylogenetic tree, but that's a great and easy way to either look at the alignment for of those isolates for this gene region, or to download a multi-FASTA of all of the isolate sequences in this gene region. So now the other way to think about this data is from the perspective of the SNP. We have a search that allows you to return SNPs regardless of its location within a gene. So let's go there. I'm going to search for SNPs based on the difference of isolates within a group. We're looking at Cryptosporidium parvum, and the data set is isolates from animals. I'm going to leave these as default parameters, and I'll click Get Answer. So now I have 4,667 SNPs that are associated with the isolates from animals. Doesn't matter whether they're in a gene or not. And if I look at this table, I can go to the SNP record page. I can tell whether it's actually associated with an ID, with a gene, and other data associated with that. This non-synonymous. So let's go to this SNP record page, and I'll highlight some information there. So the SNP record page is organized like all of our other record pages with a summary at the top and a navigation panel. I'm going to scroll down. It also has a alignment tool, and here you're not aligning the gene, but you're aligning the 60 base pairs surrounding that SNP. And then if I scroll to the genetic variation section, there's a couple of great tables that summarize the alleles and the countries that these data sets were in. And the strains and sample table is really pretty useful in transitioning into viewing in JBrowse, the SNPs. So here's an isolate. So the table lists alleles and other data associated with these samples, and the SNPs in this region. But I can use this view in JBrowse button to go directly to JBrowse to view that single nucleotide polymorphism. So here we are in JBrowse. The reference sequence is on top, and I'm looking at the aligned reads for the C320 isolate, which is one of the ones from animals. And here we have the single nucleotide polymorphism, whose page we just left. Now the advantage here is that you're in JBrowse and you have all of our data available. You can turn on tracks for other isolates. You can also look at transcriptomics or proteomics on the same data set. And you have the full complement of tools from JBrowse here, zooming in and out. and whatnot. Okay, so that is what I wanted to demonstrate in crypto. HostDB, now I'm going back to my overview of the data in the Parasite databases. HostDB received 2 RNA-seq data sets. They're associated with PlasmoDB, and they are the mouse transcriptome during infection with P. vincii or P. chabadi. Now, HostDB has all of our suite of tools, so you can look at the host data in HostDB and then compare that to the complementary on PlasmoDB. Pyroplasma, there was an important genome annotation update for Theileria parvum maguga. They used the original genome assembly, but then they reevaluated with new proteomics and RNA-seq data to reevaluate almost every gene model. So that's greatly improved annotation there. ToxoDB received 1 proteomics data set about phosphorylated proteins from infected cells. I believe this was performed in RH, but we have aligned this data and presented in ME49 and GT1. So there's actually 2 data sets from 1 study associated with this in TriTrip. And TriTripDB received 1 whole genome sequence from the rump 503 isolate, and there are more isolates from the study which will be added in a future release. So, so that is an overview of everything except PlasmoDB, right? Because PlasmoDB, like FungiDB, received a lot of data this time, and so I'm going to the news there to look at it. Wasn't useful to create a slide. So it received, 5 genome sequence and annotations, mostly from P. vincii study, but also from P. knowlesii. There are 6 or 7 RNA sequence data sets that span Berghii, Chabadi, falciparum, and vincii. 3 proteomics data sets that mainly look at the sporozoite proteome, more whole sequencing data. And then our curators have been busy adding Go terms and gene names and references to this many genes in these organisms. And so, if we have like 2 minutes, I just want to point out that that the power of our databases is to search, across databases. That's our search strategy system has that power. And so this data set here, the oocyst salivary gland sporozoite transcriptome, also has proteomics data. So if we can actually go to genes by transcriptomics, RNA-seq, and run a search on that, comparing the oocyst to salivary glands, looking for upregulated genes. And since we had proper replicates for this data set, we can look at statistics. So we find 1,654 genes that are upregulated in the oocyst compared to the salivary gland sporozoite. And from the same study, then we can intersect that data with proteomics data associated with that same study. And here it is. I'm going to search for genes that have protein products that were in this oocyst sporozoite global proteome. I'll click Get Answer. And I find 1,432 genes that had proteins expressed in the oocyst sporozoite, but only 600 of those were actually upregulated at the transcript level for the oocyst. So that's our search strategy system is a super powerful way to interrogate data on our sites. I think that's all of our time. I wonder, I don't see any questions in the panel. So if that's it, if there are no questions, we thank you very much for attending our webinar. Hopefully you will look back on it if you have questions, and you can use the Contact Us link on any of our sites to send us questions. That's it, and take care. Bye-bye.
Bye-bye, everybody.